I apologize for the abrupt ending of that last video. I had a slide out of place and it sort of stopped at a bad point anyway. So we're going to go back one slide. That white trait and all of the other traits that were lost in that F1 generation were not actually lost. They were just masked or covered up by the other trait in the cross. So in this case, the white flower was covered up by the purple flower, but it appeared again in that F2 generation. That trait that was masked and then reappeared is known as the recessive trait. So let's look at how this lost trait comes about and why we get this 3-to-1 ratio that Mendel saw for all of the 6 traits that he studied. Our book calls this Mendel's model, and it's 4 related concepts that help explain the observations that Mendel saw. First, we have a definition. That definition is alleles. So a gene is a section of the DNA that codes for a particular characteristic about that individual. Let's say that characteristic is flower color. And then there are alternate versions of those genes, slight differences in those genes that make a different appearance in the individual. Those alternative versions of genes are called alleles. Other types of alleles might be a brown-eyed gene and a blue-eyed gene, or it might be dark fur color and light fur color, or it might be pigment in the skin or lack of pigment in the skin. There are all sorts of different genes and different alleles. But having different alleles accounts for the variation that you see among individuals and all sorts of genes. So at this point, we're going to officially switch from Mendel's characters to our current term, which is genes. And we're going to switch from traits to our current term of alleles. For each gene, and I just said we were switching to genes, but it says character. For each gene, an organism inherits 2 copies or 2 alleles. Each of those alleles come from a parent. Now this has its roots in meiosis. Meiosis reduces the number of chromosomes, so each gamete is going to have 1 allele or 1 chromosome in that gamete. And then when they come together into a fertilized zygote, that individual now has 2 copies of each chromosome, so 2 copies of all of the alleles on those chromosomes. So remember that all of our somatic cells are diploid, which means they have 2 copies of each type of chromosome, which means they have 2 copies of any gene on those chromosomes, and so they can have 2 different alleles or 2 different versions of that gene. They might be the same, they might be different. But the different alleles come together in the fertilized zygote. So each parent is going to pass on 1 of their alleles to that generation. If you have 2 alleles at a particular location, or a locus is another word for the position of the gene. If you have 2 alleles at a locus and they're different, one is going to be dominant and one is going to be recessive. So we said the recessive one is the one that was lost in that F1 generation. And that is because the dominant one is actually the one that's determining the organism's appearance. So it doesn't matter if it has a dominant and a recessive. If it has that dominant allele, that's the trait that we're going to see. That's the trait that's going to determine the organism's appearance. The recessive allele is sort of a lack of physical characteristic. The recessive alleles have no effect on the appearance, in this case, of flower color. And the way that a recessive behaves really depends on the disorder. Sometimes it produces a different phenotype, sometimes it produces no phenotype. But the important thing is that recessive one is covered up and can be covered up by another allele. If we look at our chromosomes here, we have 1 chromosome that would have come from mom and 1 chromosome that would have come from dad. If we look at the purple flower allele located at the flower color gene, this locus or location on the chromosome. And this individual has a purple copy and a white copy. This is shown in the DNA sequence. So if we go all the way down to the sequence level, we see the DNA sequence here, and we see that there's a different DNA sequence in that white allele. The DNA sequence is used to produce a protein. This is going to be the same for all genes. They're used, in general, used to produce proteins, and we'll see some different things that can be produced later. But right now, we're going to say that all genes produce a protein. In this case, it is an enzyme that is going to produce the pigment for flower color. So if an individual has at least one copy of the purple flower allele, that individual is going to produce purple pigment, and it's going to be deposited on the flower. So we're going to have a purple flower. Our other allele is dysfunctional. So that white flower actually means that they're not producing an enzyme for pigment. So rather than seeing a color on the flower, we see no color, or we see white. So in this case, the white flower is actually a lack of purple pigment. This can also be called a loss of function allele. It means that this version isn't functional for its purpose, so it's not producing any flower color. All right, the last part of Mendel's model shows us how alleles and genes relate to chromosomes. And this is the law of segregation. So segregation means to separate. So in the law of segregation, two alleles, or our homologous chromosomes, are separated during gamete formation. So in meiosis 1, the homologous chromosomes are separated, and each of those sister chromatids are separated into daughter cells. So when those alleles separate, they each end up into different daughter cells from meiosis. So when our homologous chromosomes separate, we are separating our alleles. And this is why each individual only gets one copy of each allele from a parent. These cells down here at the bottom, any one of those is going to go on to be involved in fertilization, where it will combine with another gamete, and you will get back those two alleles. That was one of our beginning stages of our model here. All right, so now to Punnett squares. People always ask me if we get to do Punnett squares, and the answer is yes. We will do Punnett square practice problems in class during our in-class assignment. So I'm really just going to talk about the general idea of them now, and then we'll get to do them next class period. So Punnett squares are used to visualize the potential allele combinations that are produced from fertilization. So basically, you are doing segregation and fertilization on paper. So let's introduce some symbols here. These P's are going to symbolize our purple flower gene, so our flower color gene. And remember, we said these individuals were true breeding. So that means that they have two copies of the same allele. So the true breeding purple only has purple alleles. The white flower, the white parent, only has white alleles to pass on. The capital letter is used to signify the dominant allele, and the lowercase letter is used to signal the recessive allele. In general, I don't care what letters you use to signify dominance and recessive. I just ask that you choose ones that are easy to see the difference between upper and lowercase. Some are better than others, and that way I know that you're not making a typo, or you're not writing too small, and I can tell what letters you're using. For the record, P's are not good letters. And on paper, they all look the same. But anyway, so because each individual gets one of each of the parents' alleles, our purple flowers are going to have one copy of the dominant and one copy of the recessive. This is called heterozygous. We'll come back to that in just a minute. When that F1 population produces gametes, they are going to produce gametes that either have the dominant purple allele or the recessive white allele. So when you're doing Punnett squares, the first thing you're going to do is determine what allele possibilities the parent has. So parent number 1 is going to have one big P and one little p. Parent number 2 over here is going to have one big P and one little p. Then in the middle, you're essentially performing all of the fertilization possibilities. So the dominant allele or the dominant gamete can combine with a dominant gamete, in which you get an individual with 2 dominant purple alleles. Or you can have 2 different possibilities for the heterozygous individual. You can have one dominant from one parent and one recessive from the other, or you can have the opposite, a dominant from this parent, recessive from this one. All 3 of those possibilities are going to be purple. There we see our 3-to-1 ratio. And then this guy down here has 2 copies of the recessive allele, and so he only 1 out of 4 individuals are going to have the recessive characteristic. All right, so let's summarize some of our vocabulary. So we have our dominant and our recessive alleles. And from this information, we can infer the genotype, meaning the letters or the alleles, the genetic makeup of the individual. So this individual has 2 dominant alleles. This one has a dominant and recessive. We can also infer the phenotype. That's the observable trait or the physical expression of that trait, as opposed to the genetic makeup of that trait. Individuals that have 2 copies of the same type of allele are homozygous. Homo meaning same, zygous meaning zygote. So it had the fertilized zygote has 2 copies of the same type of allele. And that can be 2 copies of the dominant or 2 copies of the recessive. And then we have heterozygous individuals. And the heterozygote is really the one that's key in determining dominance or recessive. So in the heterozygote, the one that covers up the other trait is going to be our dominant. And the one that isn't seen is going to be our recessive. Notice that we can get the heterozygote 2 different ways. When we do Punnett square problems, you will be asked to determine both the phenotype and the genotype ratios. So on the left side here, we have the phenotype or the physical characteristics displayed in a 3-to-1 ratio. And then on the right-hand side, we have the genotype ratios. So we have 1 homozygous to 2 heterozygous to 1 homozygous. And so when you do these problems, you'll be asked to determine the ratio of the phenotype and the genotype. Notice that for those heterozygous individuals, we have 2 different possibilities for genotype, different combinations of the parents, but they produce the same phenotype. So our genotype and phenotype ratios are not always going to be the same, and that's because that heterozygous individual covers up one of the alleles. All right, so let's talk about some different types of crosses that we can do. In genetics problems, you will often be asked to determine the genotype of an individual based on some particular characteristics. But sometimes you can't tell the genotype just by looking at the individual because of those heterozygous individuals. So sometimes you might be asked to determine what the genotype of a particular individual is. If, in a practical sense, this might be for producing a particular flower for a garden store or producing a particular trait in an animal that you're breeding. And in this particular problem, we're going to perform what's called a test cross. In a test cross, you're going to cross an individual whose genotype you know with an individual whose genotype you don't know, and then look at the potential number of types of offspring, and then work backwards to the genotype. One thing about the recessive phenotype is if we know that it's recessive, we always know the genotype because there's only one possibility, and that's 2 recessive alleles. So in this case, if we have an unknown purple plant and we want to determine if it's the homozygous or the heterozygous individual, we can cross that with any white flower because we know that they're going to be homozygous recessive. So then we can make some predictions. If our purple flower is homozygous dominant and we cross it with homozygous recessive, all of our individuals should be purple. So if we perform that cross and all of them are purple, we know that our parent purple plant was homozygous dominant. The other scenario is that purple plant is heterozygous for purple. And in that case, if we cross those plants, when we do a Punnett square, we would expect that half the offspring would be purple and half the offspring would be white. So then when we actually cross those plants and do the breeding experiment, we can determine and sort of work backwards from what information we get from the cross to determine that genotype of that individual. So Mendel's work was important because he was able to show 2 fundamental principles of heredity. Now Mendel didn't actually use all of these terms, alleles and gametes and chromosomes and things like that, but once we had an understanding of meiosis and how chromosomes were separating and moving, we could link Mendel's work to the ideas of meiosis. So we talked about the law of segregation already. That's where your homologous chromosomes separate and each gamete gets 1 copy of an allele from a homologous pair. Our second law is the law of independent assortment. And this one is a little bit harder to see, but it simply refers to 2 alleles for 2 different genes on different chromosomes. So this is where Mendel started looking at 2 traits at once. So the law of independent assortment says that each pair of alleles, so let's say we're looking at flower color, separates independently of any other pair of alleles. Let's say flower position. And so the law of independent assortment refers to 2 different genes on 2 different chromosomes, say flower color and flower position. The law of independent assortment says that it doesn't matter, whether or not you get one of those particular alleles doesn't affect whether or not you get a different allele on a different chromosome. And this has to do with our chromosomes lining up at the metaphase plate in meiosis. So if we have 2 from dad on the left and 2 from mom on the right, and they separate in meiosis, you're going to get gametes that have only dad's combos and only mom's combos. But they can line up a different way, and we saw this in meiosis. You can have 1 chromosome from mom and 1 chromosome from dad line up together, and then when they separate, you now have a mixture of chromosomes that were originally from mom and originally from dad. And so at the end of meiosis, you have new combinations of chromosomes. The example that we're going to talk about here is actually seed color, yellow or green. That's shown by the Y alleles here. And then seed shape, round or wrinkled. That's shown by the R alleles here. And so this is actually showing that you can get any possible combination of Y alleles with R alleles, and getting one doesn't affect whether or not you get the other. So let's look at the law of independent assortment. Let's first look at a monohybrid cross. A monohybrid cross is when you look at an individual or 2 individuals and you study 1 trait. A true monohybrid cross is where you cross individuals who are heterozygous for a particular trait. So that would be equivalent to our F1 generation. So monohybrids are the individuals that are heterozygous So it would be these individuals here, big P, little p individuals. And a monohybrid cross is where you cross those individuals. For a true monohybrid cross, crossing two heterozygous individuals, you're always going to get a 3-to-1 phenotypic ratio. But the law of independent assortment asks us to look at whether or not two different traits are inherited together or inherited independently. And so to perform this experiment, we have to work with dihybrids. Di meaning two. So these are individuals that are heterozygous for both of two different traits. So here we have our true breeding parents, one who is yellow and round seeds, one who has green and wrinkled seeds, and we're going to cross them. The only possible gamete combination from this yellow round individual is big Y, big R. The only possible gamete combination from this green and wrinkled one is little y, little r. So that means all of the offspring are going to be big Y, little r, big R, little r. I'm sorry, big Y, little y, big R, little r. So dihybrid. They are heterozygous for two different traits at the same time. So here's our dihybrid cross. I will write this out in class. I will show you how to set this up and how to do this. But it's performed the same way as a monohybrid cross. You're first going to determine all possible gamete combinations. So we already talked about this for the F1. There's only two. And so all individuals are going to be, I'm sorry, I'm at the wrong one, are going to be big Y, big R, little r. If we are looking at independent assortment, that means that there's equal chance of each gamete getting either big Y, little y, with either big R, little r. If it was not independent, if it was dependent assortment, we would only have two options, and I'm actually going to kind of ignore that one for now. But if we determine all of our gamete combinations from this F1 generation, we see that we can get a lot of different phenotypes. We can actually get 4 different phenotypes from a variety of different genotype combinations. There are 16 possibilities. But with this dihybrid cross, where we cross two heterozygous individuals, you always get a 9-to-3 to 3-to-1 phenotypic ratio. That means that 9 of these individuals are going to be just like the big Y, big R, yellow and round parent, and then 1 out of 16 is going to be green and wrinkled. And then we have 2 new combinations here. We have a green and round, and we have a yellow and wrinkled. And that's because those gametes are assorting independently and combining to make new combinations. Make sure you remember this 9-to-3 to 3-to-1 ratio. This is really important because if you start crossing things that are not dihybrids, you're going to get different ratios here. But that dihybrid one is seen every time you cross heterozygous individuals. So the law of independent assortment is really demonstrated by that dihybrid cross. And this is important because different genes are located on different chromosomes, and individuals are a combination of all of their genetic traits. And so we can get a variety of combinations, and that's why sexual reproduction is so important and meiosis is so important.